Hello everyone, myself Dr. Vineet Kumar, Assistant Professor, Department of Zoology, the Cochin College. Welcome back, and we are now at the last session of our topic prawn structural diversity. So in this session, we are going to see the reproductive system, both male as well as female reproductive system, and the life history and development where we see the fertilization as well as the different larval stages of prawn or Penaeus. So first we'll see the male reproductive system. The male genital organs include a pair of testes which are present on both the sides of the thorax. As you see in the diagram, the testis is a tubular structure. So this particular region, this particular tubular region is called as the testis. So testis is tubular in the case of prawns. And these tubular testes, they contain many coiled structures inside which are called as the seminiferous tubules in which the sperms are produced. And as you see in the diagram, the right as well as the right as well as the left testis on both sides, they are fused or they are united anteriorly. So you can see a bridge here. So this is a bridge which connects both the right as well as the left testes. They are united anteriorly and they are free or they are separate posteriorly. Each testis is having a lateral outgrowth. So these are the lateral outgrowths from each testis, which are referred to as the testicular diverticula. Leading from the testis, you can see as they are separate on both sides, they are having separate tubes which are moving towards the posterior region. And these very highly coiled tube that is arising from both the testes are called as vas deferens, or they are called as the vas deferens, or they are called as the sperm duct. And these sperm duct or the vas deferens, they terminate into a dilated. This is the dilated terminal region of the vas deferens, and this dilated terminal region of the vas deferens is called as the ejaculatory bulb or the seminal vesicle. These seminal vesicle, they end with an ejaculatory duct which opens to outside through the genital opening or the genital pore. The genital pore is seen at the it is seen at the coxal region of the fifth periopod or the fifth thoracic appendage. So here the coxa, which is nothing but the basal region or the basal segment of the protopodite of the fifth periopod, the genital opening can be seen there. So this is the structural details of the male reproductive system. So they are having paired testes, right and left testes in the thoracic region, and they are tubular structures which is united anteriorly and they are free posteriorly. Leading from each testis, we saw there are highly tubular structures which are called as the vas deferens or the sperm duct, which ends in a gland which is called as a seminal vesicle, which is followed by a the tubular opening of the duct which is called as the ejaculatory duct, which ends in a pore which is called as the genital pore. And this opens at the fifth coxa of the last walking leg of the thoracic appendages. So this is the details about the male reproductive system. From this, or the sperms which are produced in the testes, they move through the vas deferens and reach the ejaculatory bulb or the seminal vesicle. And these sperms, the secretions from the seminal vesicle glue the sperms together and they form sperm clusters which are called as the spermatophores. And it is these spermatospores which are stored in the seminal vesicle until they are transferred to the female during mating. So now we are moving on to the female reproductive system. So female reproductive system or the system consists of the female genital organs, which include a pair of ovaries on each side of the thoracic as well as the abdominal region. Compared to the male reproductive system, female reproductive system is more elongate As a result, it covers both the thoracic as well as the abdominal region. And one difference from the male reproductive system is that, as you see in this particular diagram, the female reproductive structure, it is free or separate anteriorly, and it is fused posteriorly. It is fused posteriorly. As seen in the male, we saw some outgrowths from the ovaries or testes, which are called as the testicular diverticula. Here also you can see an outgrowth from the ovaries, both the ovaries, which is called as the ovarian diverticula. And the duct, which is called as the oviduct, through which the eggs comes out. It is seen, or it is somewhat seen in the middle of the reproductive system. The duct comes out through one of the ovarian diverticula, as you see in this diagram. It comes out through one of the ovarian diverticula, the oviduct, and the genital pore or the genital opening can be seen in the coxa of the third working leg. So it is through this particular opening at the third working leg, the ova is released from the ovaries. So these are the structural details of the female reproductive system. Next we will see the fertilization as well as the developmental stages. So fertilization is external, meaning that both the sperm and the eggs, they meet in the external environment, and development is indirect, meaning the young one which comes out of the egg, it has to pass many stages, which are usually called as the larval stages, to reach the adult form. That's why it is called as an indirect development. Mating usually occurs in the breeding season. Male deposits the spermatozoa into the thelicum of the female, which is a cup-shaped opening in the female, where the sperms are, or the spermatophores are deposited. Simultaneously, the females discharge the ova, large number of ova. Here the ova usually remain attached to the ventral part of the abdomen for some time, and fertilization occurs. So after the ova is deposited, the sperms in the thelicum, they fertilize the ova. Females carry fertilized ova till they hatch out into the first larva, which is called as the nauplius larva. So now we are going to see the 5 larval stages of Penaeus. As you can see in the slide, the 5 larval stages are nauplius, followed by metanauplius, protozoea, zoea, and mysis. So these are the 5 larval stages of Penaeus. So we are going to see the structural details of these larval stages. So to begin with, in these larval stages, what we are going to see is that we have seen the structure of an adult prawn or Penaeus, where we saw that it is having about 19 segments and 19 pairs of appendages. So in these larval stages, we are going to see such a development from an unsegmented nauplius larva to a completely segmented adult. So we are going to see these changes, how segmentation in each level segmentation occurs, how different structures develop, and in what stages what all appendages are formed, and in which stage is a complete larva which resembles the adult is formed, and these are the points which are going to be discussed under this larval stages. So we begin with the larval stages. The first one is the nauplius larva. So nauplius larva is a microscopic free swimming, which is a pelagic larva. As you see in the diagram, it is oval and unsegmented. So we are starting from an unsegmented larva, and we'll be moving on to a completely segmented adult. So this is an unsegmented oval larva, which is called as nauplius. And the features of this larva, as you see, it is having an eye, which is a median eye. So this is the eyes, the region of the eye. A median eye can be seen. And you can see some setae, which is called as the furcal setae, and 3 pairs of appendages. So this is one, this is the second appendage, and this is the third appendage. In the posterior region, you can see 2 spikes, which are called as the furcal setae. And these appendages, all these three appendages, they are also provided by locomotory setae. Most of these appendages, they are locomotory in function because of the presence of the setae. And as you see in the diagram, the first appendage, so these are the first three appendages as we see in the adult. The first one is the antenule, the second is the antennary appendage, and the third one is the mandibular appendage. These appendages in Nauplius, they do not resemble that of the adult. These are the primitive stages, or the initial or the primitive stages of the adult, which appendages which will be developed into that of the adult in the coming larval stages. So these three, as we saw, in the adult, these are mainly meant for sensory as well as for feeding. Here they are completely locomotory in function. It helps in locomotion. And the first, second and third appendages, the antennary as well as the mandibular appendages, as you can see, two branches, they are the biramus appendage. The first one, the antennary, is uniramus appendage. Mouth is, so this diagram is the dorsal view. Mouth is on the ventral side and anus is seen at the posterior tip between the furcal setae. So these are the details of the Nauplius larva, which is an unsegmented and oval larva with three appendages and a dorsal median eye. Coming to the next larval stage. So next larval stage means this Nauplius larva undergoes many molting processes, and after many molting it emerges into the next larval stage, which is called as the meta-Nauplius larva. So you can see the differences in Nauplius and meta-Nauplius in the figure itself. So the body has been elongated from the Nauplius, and it is now divisible into two regions, the cephalothorax as well as the slender abdomen, and somewhat oval structure, which is the cephalothorax, and the slender abdominal region. The cephalothorax, as you zoom into the diagram, the cephalothorax, so in the cephalothorax, you can see at the most anterior tip, you can see two structures, a paired structure, which are called as the frontal organs. These frontal organs are seen throughout the larval stages from in every from meta-Nauplius to every larval stages till the adult stage is attained. You can see this frontal organs. Frontal organs are present, and it bears the antenules, the antennae, and the mandibles, which were present in the Nauplius. And you can see the rudiments. Black dots can be seen here. So these are the rudiments of the next sets of appendages. They are the ruminants of the first, the rudiments of the first and second maxillae and the first and second maxilliped. And in this particular stage, as we said in Nauplius, all the three appendages, including the mandibles, were locomotory in function. Here, mandibles become masticatory in function. It acquires the function in feeding. So these are the details pertaining to the second larva, which is meta-Nauplius. So from Nauplius to meta-Nauplius, we saw that the differences was regarding the structure of the body. It was divisible into two regions, and also rudiments of the next four appendages also developed, and the mandibles attained masticatory function. Moving on to the third larval stage, which is protozoea. So the structure is somewhat similar to that of the meta-Nauplius larva itself. Body is divisible into both cephalothorax and abdomen, and the major change that happens in protozoea stage is that the cephalothorax is covered by the carapace, and 7 pairs of appendages are well developed now. 7 pairs in the sense the 3 of the Nauplius and 4 the first and second maxillae and first and second maxilliped, which were rudiments in meta-Nauplius, is now well developed. Thus 7 pairs of well developed appendages can be seen in can be seen in this protozoa stage, and the rudiments of the third maxillipid also develop. Another important development in the protozoa is that the lateral eyes, which will become the movable stalked compound eyes, develop. The lateral eye starts to develop on either side of the dorsal median eye, which is present in both nauplius as well as metanauplius. The median eye was present in both metanauplius and nauplius, and now in protozoa stage you can see the development of the lateral eyes on both sides of the median dorsal eye. So after many molting of protozoa, now it moves on to the fourth larva, which is called as the zoea stage. So in zoea stage, you can see more segments develop in the abdominal region. So now you can see that the cephalothorax becomes six segmented, even though there is a carapace. The cephalothorax becomes six segmented, and also abdominal segments starts to appear. Another development in the carapace is that the rostrum, the pointed structure which is called as the rostrum. As you zoom, you can see a small rostrum is being developed. So this is the rostrum, a pointed spine which is called as the rostrum develops, and the median eye disappears. In zoea, the median eye disappears, only the lateral eyes are present. So the lateral eyes were just rudiments in protozoa, now it becomes completely stalked and movable. The 5 pairs of walking legs develops as buds, and another development, as you see in this diagram, is at the most posterior region, two structures develop, which is this, the last abdominal appendage, which is the uropod, as well as the telson appear. So the main development in zoea is that both the cephalothorax as well as the abdominal segments appear. The rostrum is seen, then median eyes are disappearing, and the lateral eyes become stalked and they become movable. And the appendage which is called as uropod, and the terminal segment or the terminal plate which is called as telson also appear. So these are the major changes that you see in this particular larva, which is called as zoea. So now gradually segmentation can be seen in the abdominal region also, and now after many molts, zoea is now transformed into the last larval stage of Penaeus, which is called as the mysis. So as you can see in the diagram, mysis is somewhat similar to that of the adult Penaeus, but it is very smaller in size. The walking legs, all the walking legs of the 5 walking legs of the thorax or the cephalothorax, they become functional. The first 5 pairs of abdominal appendages develop. So we saw in the zoea that only the last abdominal appendage or the uropod were developed, but here the first 5 abdominal appendages, they develop and they become functional. After many molting processes, the mysis, they increase in size. All the appendages, they become more and more functional, and mysis, after many molts, they transform into an adult prawn. So this is regarding the larval stages. So in this session, we saw the male reproductive system, female reproductive system, followed by the 5 different larval stages, where we saw how an unsegmented nauplius larva in the series of molting into metanauplius, protozoa, zoea, and mysis, it develops into an adult Penaeus.